Good afternoon. Our project is separation of mixtures in microfluidic devices, a macroscale model using magnets.
I'm Susanna Crow.
I'm Olivia Mastrangelo.
And I'm Kanchan Railcar. Okay, so first we'll give you a little bit of background on microfluidics, and we'll talk about the gravity and water-based setups that we created, and we'll tell you about the methods of creating those, and then we'll talk about our conclusions and plans for future work.
So one of the first questions that we asked ourselves when we began this research was, why are microfluidic devices important? Microfluidic devices have surged in popularity in recent years for three main factors: their low cost, their fast reaction times, and their high productivity. Microfluidic devices are extremely small, as you can see by these measurements on this microfluidic device, and the channels within them for liquid to flow through are even smaller. So this means that very small amounts of reagent are needed for the microfluidic device to actually function, which saves costs for the small amounts of reagent. And because these small amounts of liquid are used, the reaction times in these devices are much quicker, and ultimately because of this high cost and the fast reaction times, microfluidic devices are much more productive than the traditional lab setup.
So microfluidics is an established field. There are already several ways of separating microfluids. We have centrifugal separation, which separates particles based on their density. We have deterministic lateral displacement, or DLD devices, which separate particles based on their shape and size. Filtration also differentiates particles based on their shape and size. But what's most important to know is that even though there are several kinds of devices, they all work best in different situations, separating different types of particles, so there's always room for new types of devices. You can see the deterministic lateral displacement model here and how it separates particles based on their size.
Okay, so a subset of filtration is gel electrophoresis, which separates DNA fragments based on their size and their charge. And an up-and-coming field in the medical field, a separation device that's used is magnetic tagging, which has been used in cancer research to separate tumor cells out from other body cells. So there's a diagram here of magnetic tagging.
So our research was a macroscale model of a microfluidic device, and the primary reason for this was because microfluidic devices are so small that it's difficult to observe what happens in them. And so we were able to translate the microfluidic device to a macroscale through a process known as dimensional analysis, in which the time frame and the scale of a model are increased while the concepts of physics used in the model remain the same. And here is a model, a deterministic lateral displacement device made using Legos.
So when you are designing a macroscale model of something that would actually occur on the very small scale, you have to consider how different phenomena are much more important in determining the motion. For example, Brownian motion, which is the random motion of all particles above absolute zero. You wouldn't notice it in your macroscale model, but on the microscale, it would definitely be a huge factor in influencing how particles move. Surface tension and surface to volume ratio both sort of describe how in a very tiny volume of fluid, the interactions and bonding that happen on the surface of that volume are much more influential in determining where that fluid moves rather than just the mass as a whole.
Okay. So our initial plan in sort of creating this device to separate the different size particles, we decided to take our research on deterministic lateral displacement devices, or DLD devices, and our research with magnetism. So we found that DLD devices are very effective, but their use of physical barriers to separate different size particles causes a lot of damage to the particles. So we thought about using magnets to sort of alleviate these issues with collisions. So our first preliminary setup was an 8x8 grid of strong disc magnets, and we sort of changed the angles and tried to see if we could find a pattern, but we didn't end up finding any pattern, and so this was basically just a general setup to spring off of.
After this experiment with the DLD device, we created 3 further gravity-based setups using those same disc magnets, as well as strip magnets and powerful bar magnets for certain setups. And we attached them onto acrylic boards and then used 3 different sizes of steel beads to simulate the particles in these devices. And to ensure that all of the beads entered the board from the same height and at the same velocity, we 3D printed a piece with several channels for the beads to roll down.
So after our initial DLD magnetized simulation, during it actually, we realized that the smaller beads had this tendency to get stuck on the top of the incline that we use and didn't roll all the way down. So when we designed our next setup, we decided to take that information to our advantage. and we separated the particles based on regulating the distance between the particles and the magnetic surface. So basically, you can see that we had a setup with a series of steps. So the magnetic surface was level on the bottom, and we would drop the beads down from the dispenser, and they would roll down, and what we predicted was that the smallest beads, since they had the least momentum, because momentum is mass times velocity, the smallest beads would be overcome by the magnetism because their momentum was not high enough, and they would be stuck on the highest step that was farthest away from the surface, and then the largest beads would roll down and not get stuck till they were much closer on the lowest step. So our results for the first trial, we had 3 steps, and you can sort of see in the first column that it was just way too far from the magnetic surface and nothing was stuck there, so we decided to have a trial with just the 2 shorter steps. And here you can see that it was just as we predicted. 27 out of 30 of the smaller beads were stuck on the highest step, and 26 out of 30 of the largest beads were stuck on the lower step.
For the second setup, we sort of built off of this idea of varying the magnetic field. So we used different types of magnets this time. So at the top, you can see these are weak strip magnets that were meant to catch the smallest size beads. And then in the middle portion, we put strong disc magnets to capture the medium-sized beads. And at the bottom was a powerful bar magnet, which would trap all of the largest beads. And we did 6 trials of 15 beads each. So this was something that we changed from the first setup to avoid issues that we found with clumping. And this is just a picture of the actual setup 2. This setup ended up being extremely effective. So all 30 of the small beads that we dropped landed in the weak strip magnet area at the top. 21 out of the 30 medium-sized beads landed in the middle section on top of the strong disc magnets. And 20 out of the 30 large beads were trapped on top of the strong bar magnet at the bottom.
And for our third setup, we experimented with the effective spacing of similar strength magnets. And so what we did is that for the setup, we had a row at the top of magnets that were spaced twice as far as the magnets in the other 2 trials, so they were spaced 5 centimeters apart. And then 4 rows of magnets that were spaced the same as the other 2 trials, so 2.5 centimeters apart. And then at the bottom, a row of closely spaced magnets that were 1.25 centimeter apart. And like setup 2, we did 6 trials of 15 beads per trial to avoid clumping. And we hypothesized that the smallest beads would settle in the areas with the widest space because they had less momentum to overcome the magnetic force, but the largest beads, which had more momentum, would not stop until the very bottom set of closely spaced magnets where the magnetic force would overcome their momentum. However, this hypothesis turned out to be incorrect. As you can see, 17 of the small beads ended up in the middle section instead of the wide space section at the top. The medium-sized beads were the only beads that met our hypothesis, with 19 out of the 30 ending up in the standard space section. And the large beads were also unsuccessful, with 13 ending up in the closely spaced section, but 13 also ending up in the standard space section.
So once we conducted our 3 gravity-based trials and concluded that setups 1 and 2 were the most effective, we decided to translate it into using a flow of water as the driving force rather than gravity, because it would better simulate what happens in a microfluidic environment. So for our 4th setup, we basically had the setup in a shallow tank here, and the flow of water came from a water jug that was placed above it, so it would create a higher flow, and then it would continue out through tubes to this dispenser here. In setup 4, for this tank, we had sort of an inverse of the gravity-based trial. So here, that magnetic surface was tilted underneath a level surface that the beads would travel on, and again, we expected the smaller beads to stop sooner when the magnetic surface was farther away, and the larger beads to stop later when it was closer.
So this 5th setup was similar to setup 2 with the varying strengths of magnets. And the thing we changed in this 5th setup was how we got the flow of water to the actual tank with the device in it. So the first thing we did was the same as in setup 4 with the water jug, which was at a little bit of a higher point than the tank. But we found that the velocity of the water coming into the tank was not enough to effectively push the beads. So we tried putting the water jug at the top of a staircase, which was about 5 meters high, and that allowed for the flow rate to increase, but there were some issues with the connections of the tubes that kind of inhibited this. And then we tried using a pump, which would have worked because it was useful in controlling the flow rate, but we had issues with priming the pump, and there were a lot of air bubbles in the tubes, so that was causing some problems. So our final setup involves a direct flow of water from the sink, and that was the best one that we found because it was easy to fluctuate the flow rate using the faucet, and it was a pretty compact setup. So this is just a diagram of the tank with the device right here, and then this is just tape and rubber and plastic to fill it up and decrease the cross section to create a greater flow rate.
And here is a picture of our model with the tank 5 meters above the flow. As you can see, this is the length of plastic tubing that we used. And if you can see, we had some problem connecting the tubing, which led to an unsuccessful flow rate and lots of leaking. And then here was our final setup with the sink. Here's the tube that's connected directly to the sink. And here's the setup clamped to the counter.
So we definitely had our share of technical difficulties in relation to the water-based setups. First off, for setup 4, when we needed an incline, the tank was too shallow to really accommodate the angle we needed. All the small rubber and plastic components that she mentioned created, they didn't fit perfectly, so there were air bubbles and air pockets that would interrupt the flow. And as she was saying, we really had to experiment with how to create a flow of water because the original tank was too low to generate the flow rate we needed. So unfortunately, we couldn't fabricate a working setup with a water-based model. When we finally did, as she was speaking about, when we attached it to the sink and we did create a flow of water that was strong enough, the setup couldn't support it and started leaking excessively. But in that short amount of time, this picture here, you can see other issues where the beads aggregated near the entry point. They didn't spread out all at once as we sort of hoped they would. And our small triumph is that we noticed that the smaller beads did not travel as far as the larger beads.
So our ultimate conclusions in this experiment were that with the gravity-based setups, the setups in where the distance between the magnetic field and the beads varied were the most effective setups, as in setup 1, where there was physical distance between the magnets and the beads that strengthened and weakened the magnetic force as it went along, or in setup 2, where various strengths of magnets were actually used. And through our experiments with the water tank, we did learn that it more closely mimicked a microfluidic device than our gravity-based setups because it had the added flow. However, we were not able to successfully complete enough trials with that to get enough data. But we did find that magnetism is a viable way of separating particles based on size in microfluidic devices.
So in the future, if we continue to pursue this project of making a macroscale model, we would try to find a larger tank, first of all. We would probably get more custom fit lining and so that we wouldn't create so many air bubbles and interrupt the flow as much. Just in general, improve the setup so that we wouldn't have as many malfunctions.
And if further research was done, we would hope that our experiments would be used in the real world, such as mentioned before, with magnetic tagging, and other research that we found showed that DLD devices were used to separate blood cells, so hopefully our research could be used in the future for something like that.
And there are a number of people who we would like to thank for their contributions to our project. First and foremost is our RTA, Laura Gunderson, for all of her unconditional support and guidance throughout our research.
We'd also like to thank our project coordinator, Professor Armand Drazer, for really getting us started and helping us through the process of our project. And we'd like to thank the graduate and undergraduate students working in Professor Drauzer's lab because they were really helpful, especially with the water-based setups.
And of course, we'd like to thank people who made GSET possible, the director, Eileen Rosen, and the assistant director, Jean-Patrick Antoine. And of course, we'd like to thank Rutgers for hosting Governor's School.
And lastly, we would like to thank the state of New Jersey, as well as our sponsors, Morgan Stanley, NJ Resources, South Jersey Industries, and PSE&G for their continued support of the Governor's School program. If you have any questions, we'd be happy to answer them now. Thank you.
microfluidic devices are established technology, and I was just glancing at the paper, they're mostly used in labs right now, or are they making their way to some application in my house sometime soon?
So microfluidic devices, a large part of them is what's known as lab-on-a-chip technology, where several processes can be run on one chip to simulate a lab on a microscopic scale. So they can be found in places such as inkjet printers and DNA chips and glucose meters. So those, not all of those might necessarily be in your house, but they are technology that is out there.
How do you magnetically charge a cell or any particle?
I'm pretty sure of that. Okay, it's done mostly like in a lab. Those tests. So, okay, yeah. So they basically just have to use magnetic metallic substances to tag certain cells, and then they would sort of send it through a device that has like speed bumps. So it would sort of push these magnetized particles off to the side, and then the unmagnetized particles would sort of keep going through the flow.
Any other questions?
Thank
you very much.
Thank you.
for listening.